By the end of this hour, you are going to understand why your nervous system is essentially a tube that got incredibly complicated, how a group of traveling cells are responsible for building your face, and why a simple vitamin is the most powerful tool we have to protect this entire process.
It's a journey from a flat sheet to a 3D command center. It's pretty amazing.
Welcome back to the Deep Dive. Today we are tackling one of the most, I think, dramatic physical transformations in all of human existence. We're looking at the Great Fold.
It really is a massive moment. I mean, if you visualize the embryo before this all starts, we're talking early in the third one, it's basically a flat, three-layered disc.
Like a little pancake.
Exactly, like a pancake floating in fluid. But by the time this is all over, that pancake has rolled itself up into this complex, tubular, three-dimensional structure.
And the stakes here couldn't be higher. We're not just making a tube like a garden hose. We're talking about a flat sheet of cells that is deciding to become the command center for the entire body.
The whole thing, brain, spinal cord, peripheral nerves, it all happens right here, right now.
So this is the moment complexity just accelerates.
It does, and if this fold goes wrong, the consequences are catastrophic. We're talking about conditions that are either incompatible with life or cause severe lifelong disability. But when it goes right, it's just structurally beautiful.
Okay, so to guide us through this whole folding process, we're pulling from our stack of 3 heavy hitters in the world of embryology.
The usual suspects.
First up, we've got Langman's medical embryology.
Right, and Langman's is just fantastic for that deep molecular mechanistic explanation. It tells you how it works, all the signaling pathways.
And we're balancing that with Moore and Prasad's The Developing Human.
Moore is our clinical lens. It's the one that connects the embryo to the patient you might see in the clinic. It perfectly answers that "so what" question.
And rounding out the trio, we have IB Singh's Human Embryology.
IB Singh is our roadmap. It's great for the structural framework. It simplifies the sequencing, and it helps you keep your orientation when things start, twisting and turning.
Perfect for exam prep.
Exactly. But as always, we have to start with our disclaimer.
Of course.
This conversation, it's an interpretation. It's a synthesis of these texts. We're trying to spark your curiosity and help you grasp the underlying logic.
Correct. This is a guide. It's a revision aid, but it is not a replacement for actually reading the books. Please, keep your textbooks open while you listen.
Definitely. You'll want to be looking at the chapters on the nervous system, the derivatives of the ectodermal germ layer, and the 4th to 8th weeks of development.
Seeing the diagrams that Langman or IB Singh have while we talk, it just changes everything.
It really does.
So, our mission today is pretty specific. We want to move beyond just memorizing days and folds. We want to understand the logic of this process of neurulation. We're going to look at shaping, closure, and timing.
We're going to peel back the layers and see how the body literally builds its own wiring from scratch.
Okay, let's get into it. Section 1, the setup, starting from what you call the default state.
Right. Let's set the scene. We're in the gastrula stage. Moore gives us a great picture of this. The embryo is a trilaminar disc. 3 layers.
Ectoderm on top, mesoderm in the middle, and endoderm on the bottom.
And we are laser focused on that top layer, the ectoderm.
When I think of ectoderm, my brain immediately goes to skin, epidermis, the stuff on the outside.
And that's a fair association because that's where most of it does end up. But here is the twist that Langman reveals, and it's absolutely fascinating. It turns out the default state of the ectoderm is to become neural tissue.
Wait, what?
It wants to become the brain.
So you're saying if I took a generic ectoderm cell, put it in a dish all by itself with nothing else around, it would become a neuron?
In theory, yes. With no other signals, its default pathway is to differentiate into neural tissue.
So, why aren't we just giant walking brains? What stops the whole sheet from becoming a brain?
An inhibitor. There's an molecule called BMP4, bone morphogenetic protein 4. This protein just floods the ectoderm, and it's basically shouting, no, be skin, be epidermis.
Oh. So BMP4 is the signal that overrides the default.
It is. It effectively ventralizes the ectoderm, telling it to form skin. So for the brain to form, we don't need a signal that says be brain. We need a signal that stops the be skin signal.
We need to block the blocker.
We need a rescue team. And this is the molecular switch Langman describes so beautifully. The primitive node, the notochord, and the prechordal mesoderm, these structures right along the midline, they start secreting these superhero molecules.
I love a good superhero list. Who are they?
Noggin, chordin, and follistatin.
Can't make that up. Noggin. A molecule that helps make your head is literally called noggin.
It's perfect, isn't it? So these three, noggin, chordin, and follistatin, are inhibitors of the inhibitor. They physically bind BMP4 and block it from working.
So by blocking the signal to B skin, they allow that patch of ectoderm to default back to its original destiny.
Which is to become neural tissue, and that whole process is called neural induction.
And you mentioned another signal, FGF.
Yeah, fibroblast growth factor is also in the mix here. It helps upregulate those inhibitors and also represses the production of BMP. It's like a reinforcement loop, making sure that midline stays protected.
Okay, let's get a visual anchor here for everyone listening. Imagine a big flat sheet of cells out in the rain.
Okay.
The rain is BMP4. Everywhere the rain hits, the cells become skin. But right along the midline, the notochord is holding up a big umbrella. That umbrella is noggin and chordin.
I love that.
The dry part under the umbrella becomes the neural plate.
That is a brilliant analogy. And physically what you see is this thickened, slipper-shaped plate of neuroectoderm appearing right down the middle of the dorsal surface. The cells actually get taller. They look different. That's your neural plate.
Okay, so we've got our plate. But a flat plate isn't a nervous system. We need to solve it. Section 2, neurulation. The mechanics of folding.
Right, and this is where IB Singh and more really help us with the sequence. It's a clear progression. You start with neural plate, that thickened zone we just made.
Day 18 or so.
Around day 18, yeah, the plate starts to invaginate right along its central axis. It sinks down.
Forming the neural groove, like a valley down the middle.
Precisely. And as the middle sinks, the edges rise up. Those are the neural folds.
And the goal of the folds is to meet in the middle and fuse.
Exactly. And when they fuse, you have your neural tube.
It sounds so simple. Plate, groove, folds, tube. But Langman points out something critical about the shape. It's not just folding. The plate itself is actively getting longer.
Yes, convergent extension. This is a vital concept and easy to miss. As the neural plate is forming, the cells in it are actually moving. They're moving from lateral to medial.
So they're squeezing in toward the midline.
They're squeezing in, which makes the plate narrower, but much, much longer.
Why that matters so much?
Because this lengthening is what drives the elongation of the entire body axis. If the plate just stayed short and fat, the tube couldn't form correctly. The geometry would be all wrong.
And there's a specific genetic pathway for this, right? The planar cell polarity pathway.
Correct. It's regulated by genes like VNL, and we know this because if that pathway fails, the plate stays short and wide, and the tube can't zip up properly. It's a mechanism we see failing in certain neural tube defects. It shows you that the shaping is just as important as the folding itself.
Speaking of zipping up, let's talk about that closure. I used to picture it just snapping shut all at once, like closing a book.
A common misconception. It's not like that at all. You should think of a zipper.
Okay.
But it's a weird zipper. The fusion begins right in the middle in the cervical region around the fifth somomite, and then it zips in two directions at the same time.
Up towards the head?
Cranially, yep. And down towards the tail, caudally.
So for a while, the tube is actually open at both ends.
That's right. These openings are called the neuropores. The anterior, or cranial, neuropore at the head end, and the posterior, or caudal, neuropore at the tail end.
So the inside of the developing nervous system is open to the amniotic fluid.
For a few days, yes. The amniotic fluid is flowing right through the future spinal canal. The embryo is an open system for a bit.
And the timing on this is absolutely critical. This is high yield for exams, but also just for understanding the clinical picture.
It is. The anterior neuropore, the one at the head, closes around day 25. That's at about the 18 to 20 somite stage.
In the posterior one.
The posterior neuropore closes later, around day 28. That's the 25 somite stage.
I have a really simple mnemonic for that. 25 comes before 28. The brain shuts the door before the tail does.
That works perfectly. And keep that day 28 number in your head. It's the end of week 4. That timing is going to come back to haunt us when we get to preventing these defects.
Right. And while the zipper is moving, Moore reminds us we can't ignore the bigger picture. The entire embryo is folding.
Oh, absolutely. While the ectoderm is rolling up on top to form the tube, the whole disc is curling in on itself.
So you have the head fold.
Right, which pushes the developing heart and the oropharyngeal membrane down into the ventral surface. It's basically moving the heart into the chest.
And the lateral folds are curling the sides in.
Exactly, creating a cylinder. And in doing so, they're trapping the endoderm inside to form the gut tube.
So it's a total remodeling job. The flat disc becomes a tube within a tube.
Okay, let's just pause for a second. Can you really visualize that zipper moving in two directions from the middle?
Yeah.
Now imagine what happens if the top zipper gets stuck versus the bottom zipper. Just keep that image in your mind, because when we get to the clinical section, that is exactly what determines the type of defect we see.
Before we get there, though, we have to talk about my favorite group of cells. Section 3, the neural crest.
Ah, yes, the so-called fourth germ layer.
Why do they call it that? I thought we only had 3, ectoderm, mesoderm, endoderm.
Well, they're technically ectoderm. They originate from the very edges of the neural folds. But they are so distinct, so versatile, and so important that many embryologists just treat them as a whole separate category.
So they're located right at the crest, the peak of the neural folds.
Right as the folds are about to touch and fuse, these cells are sitting right at the very top.
And then they do something pretty remarkable.
They do. They undergo EMT epithelial to mesenchymal transition.
Can you break that down for us?
Sure. Think of epithelial cells as being like bricks in a wall. They're stuck together. They form a barrier.
Okay.
Mesenchymal cells are like independent travelers. They're migratory. They can detach and move through the tissue on their own. So these cells basically quit their job as bricks and decide to go exploring.
What drives that? Why do they suddenly decide to leave home?
It's all about the molecular gradient again. Remember our umbrella analogy?
High BMP on the skin side, low BMP under the umbrella on the neural plate side.
Exactly. The neural crest cells are sitting right at the border of the umbrella's shadow. That intermediate level of BMP signaling induces a very specific cascade of genes.
And Langman lists them out for us. PX3 induces SNAIL and FOXD3, which specify the cells as crest cells.
Right. And it also induces SLG, which promotes their migration.
SLG is the one that tells them to pack their bags.
It is. It represses the molecules that hold cells together, like E-cadherin, and basically allows them to slip away from the closing neural tube.
And where they go?
Pretty much everywhere. They are the great explorers of the embryo. In the trunk, Langman describes 2 main pathways. There's a dorsal pathway.
Dorsal, so towards the back.
Correct. These cells move through the dermis and they become melanocytes.
The pigment cells in our skin.
Yes. The reason you have skin color is because these neural cells migrated into your skin back in the 4th week of development.
That is incredible. And the second pass.
The ventral pathway. These cells move ventrally through the anterior half of the somites, and their derivatives are just astonishing.
Give us the list.
Sensory ganglia, like the dorsal root ganglia, sympathetic and enteric neurons, Schwann cells, which myelinate our nerves, and even the cells of the adrenal medulla.
Wow. So skin pigment, the entire peripheral nervous system, gut regulation, the fight or flight response, all from the same starting cell population.
And we haven't even mentioned the cranial neural crest. These are the ones that leave the head region, and they do it before the tube there is even fully closed. They are absolutely critical for building the craniofacial skeleton.
So without these neural crest cells, we literally don't have a face.
Correct. They form the pharyngeal arches, which go on to build the jaw, the tiny bones in your ear, all the connective tissue of your face. IB Singh gives us that great rapid-fire list.
Dorsal mass gives you dorsal root ganglia.
Ventral mass gives you the sympathetic ganglia and the chromaffin cells of the adrenal medulla, but also odontoblasts for your teeth, parafollicular cells in the thyroid, the pia and arachnoid layers of the meninges. The list just goes on and on.
Okay, let's do a quick fictional vignette. Imagine a patient walks into your clinic. They have a white patch of hair right at the front, white forelock. They have 2 different colored eyes heterochromia, and they have congenital hearing loss.
Oh, that just screams a neural crest defect. That clinical picture sounds a lot like Waardenburg syndrome.
So walk us through why those specific symptoms point to the neural crest.
Well, the travelers, the melanocytes, didn't make it to that patch of hair or to one of the irises. Hence, no pigment.
And the hearing loss.
Neural crest cells are also critical for the proper development of the inner ear. So if they don't get there or don't function correctly, you get deafness. It's a perfect illustration of how far these cells travel and what happens when they get lost along the way.
It really emphasizes that embryology is dynamic. It's movement. It's migration. It's not static.
Not at all.
Okay, let's go back to the tube itself now that the travelers have left. Section 4. The architecture of the tube.
So the tube has closed. Now it needs to get organized. And IB Singh explains this really clearly. The wall of the tube differentiates into 3 distinct layers.
From the inside out.
From the inside out, yes. The innermost layer is the ependymal layer. These cells will line the central canal of the spinal cord and the ventricles of the brain.
Okay, middle layer.
That's the mantle layer. This is the crucial one. These cells become the neuroblasts and the glioblasts. This layer becomes the gray matter.
All the cell bodies of the neurons.
Exactly. And then the outermost layer is the marginal layer. This contains all the nerve fibers, the axons that are growing out from the cell bodies in the mantle layer. This becomes the white matter.
That makes perfect sense. Marginal is on the edge, white matter on the outside, gray matter on the inside. That's the spinal cord.
That's the spinal cord. But how does the tube know which side is for sensory information and which is for motor commands?
The sulcus limitans.
The sulcus limitans. It's this longitudinal groove that appears on the inside wall of the neural tube. You can think of it like a border fence that divides the tube into two functional zones, a dorsal half and a ventral half.
And this is the logic that stays with you for your entire medical career. This is the key to all of spinal cord anatomy.
It is. The alar plate is dorsal, the back half. This entire region is dedicated to sensory input.
My mnemonic for that is alar sounds like wings. Wings are on your back, they're dorsal. So alar equals dorsal equals sensory.
That's a great one. And the basal plate is ventral, the front half. This is dedicated to motor output.
Basal makes me think of base, foundation, action. So basal equals ventral equals motor.
And that fundamental dorsal sensory ventral motor split persists for life. When you look at a cross-section of the adult spinal cord, the posterior horns are sensory. They came from the alar plate. The anterior horns are motor. They came from the basal plate.
So if you're a radiologist looking at an MRI of a spine right now, you are looking at the direct result of the sulcus limitans appearing back in week 4 of development.
That is exactly right. The blueprint is laid down early.
Now, at the cranial end, the head end, the tube doesn't just stay a simple tube.
Right.
It blows up like a balloon.
It does. It forms the brain vesicles. Initially, you get 3 primary vesicles, the prosencephalon, or forebrain, the mesencephalon, or midbrain, and the rhombencephalon, the hindbrain.
But it doesn't stop there. As IB Singh details, those 3 divide further into 5 secondary vesicles. The complexity just keeps increasing.
The prosencephalon, the forebrain, splits into 2. The telencephalon, which becomes the huge cerebral hemispheres, the part of the brain you think with, and the diencephalon, which becomes the thalamus and hypothalamus, all the deep relay centers.
What about the mesencephalon?
The midbrain stays the midbrain. It doesn't split. It acts as a bridge.
And the rhombencephalon at the back?
That splits into the metencephalon, which forms the pons and the cerebellum, and the myelencephalon, which becomes the medulla.
So 3 becomes 5. Let's do a quick check-in question for you. If you look at a cross-section of the spinal cord today, can you trace the sensory horns back to the alar plate and the motor horns back to the basal plate? If yes, you've got the architecture down cold.
Okay, now for the part we've been alluding to. We have to talk about what happens when this elegant, beautiful folding process goes wrong. Section 5, clinical relevance. When the zipper fails.
Neural tube defects.
NTDs.
Right. These are simply failures of the neural tube to close. And remember our zipper analogy. The location of the failure is everything. Okay, let's start at the top. If the anterior neuropore fails to close by day 25, what happens?
You get anencephaly. The brainstem might form, but the forebrain, the cerebral cortex, the thinking brain, it never develops. Because the neuropore stays open, the neural tissue is directly exposed to the amniotic fluid and it just degenerates.
And the skull doesn't form over it.
The skull vault fails to form as well, so the rudimentary brain tissue is exposed. This condition is invariably lethal. These infants are often stillborn or die within hours or days of birth.
A complete tragedy of cranial closure. Now, what if the zipper fails at the bottom, the posterior neuropore, on day 28?
Then you get some form of spina bifida, which literally means split spine. And this exists on a spectrum from very mild to very, very severe.
Let's run through the types that Moore and IB Singh defined for us, the mildest form.
That would be spina bifida occulta. Occulta means hidden. In this case, the neural tube itself actually closes just fine. But the bone, the vertebral arch, doesn't fuse over it.
So the only sign might be on the skin.
Often, yes. A little hairy patch, maybe a dimple or a birthmark on the lower back. It's usually asymptomatic and found by accident on an X-ray.
Okay, stepping up in severity, we have meningocele.
Here, the bone is open and the meninges, the coverings of the spinal cord, protrude out. It forms a cyst on the back that's filled with cerebrospinal fluid, CSF. But the spinal cord itself is still safely inside the spinal canal.
And then the most severe form that's compatible with life.
That's myelomeningocele. Here, the bone is open, the meninges protrude, and the spinal cord tissue itself herniates out into that sac.
So the nerves are displaced and damaged.
Almost always. This usually causes severe nerve damage, paralysis, loss of sensation, bladder and bowel problems below the level of the lesion.
And there's one more, the most severe of all, rheschisis.
This is a total failure of folding in a region. The zipper never even started to move. The neural tissue lies flat and completely exposed on the surface of the back.
Now, here is where this deep dive becomes incredibly practical. Langman and Moore both put a huge emphasis on the folate story. This is something every single person listening needs to understand.
It is truly one of the greatest public health victories of the 20th century. We now know that 50 to 70% of these devastating neural tube defects are preventable.
The magic molecule is folic acid, vitamin B9. Why does it work? What's the mechanism?
Folate is absolutely essential for DNA synthesis and cell division. Now think about what's happening in the embryo during week 3 and 4. The neural folds are elevating and fusing. This requires massive, rapid cell division.
A huge amount of growth in a very short time.
Exactly. If you don't have enough folate, the cell cycle slows down. The cells just can't divide fast enough to close that gap before the developmental window of opportunity slams shut.
The recommended dose is 400 micrograms daily for women of childbearing age. But there is a massive timing issue here, isn't there?
It's the whole crux of the problem. We said the tube is fully closed by day 28.
4 weeks pregnant.
Right. And most women don't even know they're pregnant until they miss a period, which is often after day 28.
So if a woman waits until she gets a positive pregnancy test to start taking her prenatal vitamins.
For the neural tube, it's very often too late. The zipper has already failed. The damage is done before the intervention can even begin.
That's just a terrifying mismatch between our biology and our awareness.
It is. Let's do a quick clinical scenario. A couple comes in for a preconception consultation. They're just thinking about trying to have a baby in the next year. This is your moment as a clinician.
Right.
You have to explain to them clearly why the prescription for folic acid starts now, not when they get pregnant, months before. You're trying to build up their folate stores to race against that day 28 deadline.
You need the levels to be high before conception even happens.
It creates a buffer. And it's exactly why many countries have mandated grain fortification, adding folate to bread, flour, and cereal. It protects the pregnancies that aren't planned. It's a population-level solution to a biological timing problem.
Before we wrap the clinical section, just a quick mention of 2 other anomalies IB Singh highlights. First, encephalocele.
That's a herniation of brain tissue through a defect in the skull. So you can get a cyst on the head, usually the back, that contains actual brain matter. It's like a meningocele, but for the cranium.
And holoprosencephaly.
This is a failure of the prosencephalon, the forebrain to divide. Remember we said the forebrain splits into 2 hemispheres.
Telencephalon and dienencephalon.
If that splitting fails, the forebrain can remain as one single midline vesicle. In the most severe cases, this influences the face so profoundly that you get cyclopia, a single eye in the middle of the face, a devastating midline defect.
So we have covered a lot of ground. Let's try to bring it all home in the outro.
It's been quite a journey, hasn't it? We started with that simple flat sheet of cells.
A pancake.
A pancake, yeah. We watched it get rescued from becoming skin by Noggin and Torden. We saw it lengthen and narrow through convergent extension.
We watched the zipper move up and down, racing the clock to get closed by day 25 and day 28. And we saw those incredible neural crest travelers take off to build the face, skin pigment, and the entire peripheral nervous system.
And finally, we saw the tube itself organize into the LR plate for sensory input and the basal plate for motor output, the fundamental blueprint of our nervous system.
So why does all this matter? I mean, really? Because embryology isn't just history. It is anatomy explained.
It's the why. When you're in anatomy lab struggling to remember why sensory nerves enter the back of the spine and motor nerves leave from the front, you don't need to just rote memorize it. You just need to remember the alar and basal plates from week 4. The logic is hardwired into the development.
And I want to leave you with a final provocative thought to mull over. We talked about the neural crest, that 4th germ layer. From an evolutionary perspective, these cells are a complete game changer. They allowed vertebrates to develop a distinct head with a jaw, with complex sensory organs, with a face. Some evolutionary biologists argue that the neural crest is what allowed for a predatory lifestyle. To give us the tools, the eyes, the jaws, the advanced sensory processing to actively sense prey and eat it. Without the neural crest, we wouldn't have that active sensory-driven head that defines us.
So without those little travelers migrating back in week 4, humans wouldn't be humans.
We'd just be filter-feeding tubes floating in the ocean.
Wow.
On that note, here's your call to action. Open up IB Singh to the chapter on the nervous system. Find a diagram of a cross-section of the spinal cord. Look at the gray matter horns, and trace them back in your mind to the alar and basal plates. See that history looking right back at you.
And please, take your vitamins.
Thanks for listening to The Deep Dive. We'll see you in the next stage of development.